Hello, everyone. Welcome to the webinar, University of California, San Diego and Collaborating Sites Data Sources. The webinar is being recorded and will be posted on the GSA website. A notice to all attendees will be distributed once the recording is available. Today's program is the fourth in a series of 5 webinars on HIV and aging data access, availability, and research funding opportunities. For information about this webinar series, go to geron.org/webinars. A question and answer session will immediately follow the live presentation. We will be accepting questions through the questions feature accessible on the GoToWebinar panel. Also located there are downloadable handouts for today, a copy of the presentation slides. Without further delay, I'll hand the microphone to Dr. Bob Heaton, Director of the HIV Neurobehavioral Research Center.
Thank you, Judy. On behalf of my colleagues, we appreciate the opportunity to describe to your group the resources that are available in 5 major research projects that are being coordinated or conducted at the HIV Neurobehavioral Research Program, or HNRP, at the UC San Diego. The speakers, I'll begin, and then my colleagues Jenny Uticello, David Moore, and Scott Latinder will talk about other of the projects. So next, please. These are down below the 5 major studies that we'll be talking about today. Not all of them were specifically designed to investigate aging in HIV, but all have substantial numbers of older people with HIV, and in some cases HIV uninfected controls. I'll be talking about the HIV Neurobehavioral Research Center, or HNRC. Then Jenny will describe the Translational Methamphetamine AIDS Research Center, or TMARC. David Moore will describe the National NeuroAIDS Tissue Consortium, which is a national brain bank study, and also a successful aging among HIV infected adults study. And last but not least, Scott Latinder will tell us about the CNS HIV antiretroviral therapy effects research, or charter cohort study. Next. Next, please. This slide shows the evolving age distributions of HIV infected people in the US from 2000 in the purple bars to 2016 in the red bars. On the x-axis, you see the 6 major age groups going from less than 25 years old on the left to greater than or equal to 65 on the right, and you can see that the epidemic in the US is aging progressively due to greater longevity of people with HIV infection on their more effective antiretroviral therapies. Next. So I'll be talking about the HNRC next. This is a P30 grant that has as its mission to stimulate multidisciplinary neuro-HIV research at all levels, local, national, and international. Our interdisciplinary research team serves as a resource for consultation and training. And we facilitate research by identifying important questions to be addressed, and by providing access to stored data and biological specimens from our participants. And we're also able to recruit new participants to meet the specific needs of studies that we support. Next. The HNRC cohort is probably the most important resource that we have, and it importantly includes both HIV infected and uninfected adults. Next. This slide describes the 1,212 HIV uninfected on the left and infected on the right people that the HNRC has given thorough neuromedical and neurobehavioral evaluations to over the last 5 years of funding. The two cohorts are subdivided by age, less than 50, younger, and 50 plus, older. And you can see that the majority are actually in the older groups. There are more males in the HIV infected groups, consistent with the prevalence in the population in this country. There's a good mix of race, ethnicities. Substantial numbers of infected and uninfected people have had 2 or more comprehensive HNRC visits, so that they have both data and biological specimens that are longitudinal available. There are both HIV and age effects on employment, on prevalence of lifetime major depressive illness and lifetime substance use disorders, and also in prevalence of neuromedical multimorbidities, which includes things like diabetes, hypertension, cardiovascular issues, hepatitis C co-infection, chronic lung disease, and so on. Next. This slide shows the HIV disease and treatment characteristics of our younger and older infected subgroups. You can see, and not surprisingly, that the older subgroup has had much longer HIV infection. They've been exposed to ART for a longer period of time. They have more history of severe suppression in the past and historic AIDS. But importantly, both groups are quite well treated on their antiretroviral therapies. Next. This is a very busy slide, and it shows the details of the neurocognitive assessments that's part of our standard evaluation at the HNRC. All our slides are available to your group. We're happy to share them with you, and we're happy to answer questions later if you have them. But briefly, these tests cover 7 major ability areas that are recommended for classification of HIV neurobehavioral, HIV neurocognitive disorders. And they include verbal fluency, executive functions, attention, working memory, learning and delayed recall, speed of information processing, and complex motor skills. In the center column here, you see that several of our cognitive tests covering most of the major domains have been adapted for remote administration during the COVID-19 pandemic. And on the right, you can see that virtually all of the tests have been translated and adapted for administration to monolingual Spanish speakers. And importantly, we have published demographically corrected norms for all of these tests for both English and Spanish speakers. The corrections are for age, education, sex, and race, ethnicity. Next. We also have very detailed behavioral assessments beyond cognition and assessing psychiatric and emotional functioning, social support and social functioning, substance use, including their lifetime history and current history of substance use disorders, and timeline follow back substance use histories. We assess for adequacy of everyday functioning, including independence in activities of daily living, difficulties with cognition in day-to-day life, changes in employment, and overall quality of life. We assess for HIV transmission risk behaviors and some positive psychiatry features that may help mitigate problems that many HIV-infected people experience. Next. This shows our standardized neuromedical evaluations. They include detailed medical history with ICD-9 codes, medication history, and adherence to antiretroviral therapies, the standardized physical and neurological examinations. We collect biological specimens, blood, urine, CSF, and saliva and stool samples for microbiome assays. We do a urine drug screen and breathalyzer on the day of testing. We have a number of standard biomarkers that we assay for, but the neuromedical group is capable of many more assays for supported studies if they can be afforded. We have the data and we've combined the data into a number of major composite indices for stroke and heart disease risk, the risk for all-cause mortality, frailty, and comorbidities. We assess for use the Pittsburgh Sleep Quality Index for sleep disorders, and we have assays for bacterial populations in the gut microbiome, and we're beginning to do metabolic analyses to get at the functioning of the gut microbiome. Next. Our neurovirology and biology group is capable of these many advanced assays. These are done on a study-specific basis and typically not done on all of the participants in the HNRC, but they're available and the stored samples are available. Next. This shows that over the last 5 years of funding, the HNRC has supported 130 funded studies, and there are 17 proposals under review that we've pledged to support if they're successful. Next. And this shows that during the same time period, 5 years, we've mentored 178 trainees at all levels, from undergraduate to junior faculty members, including 27 international trainees. Next. This map shows our national collaborations around the country, and all the places where there are stars have our standardized neurobehavioral and neuromedical procedures in use, so that their data can be compared and combined with ours. Next. And finally, this shows the same thing internationally. And you can see there are a lot of stars around the world where we have worked with colleagues in these other countries to administer our standardized procedures. And in many of these places, we have helped the local collaborators to collect data and develop population-specific and language-specific normative standards that correct for age, education, and sex, just like ours do. Next. And now I'm going to turn it over to Jenny Udicello, who will tell you about the TMARC.
Yes, thank you, Bob. Good morning, everyone, and thank you for the opportunity to present some information and available resources from the Translational Methamphetamine AIDS Research Center, or TMARC, which is funded by the National Institute on Drug Abuse. My name is Jenny Udicello. I'm an assistant professor at UCSD, and I served as a co-investigator on the TMARC alongside Drs. Heaton and Latender, who co-directed TMARC's primary clinical core. So as the NIDA-funded P50 Research Center, the TMARC served two primary roles. One was to conduct research, which was a primary function of the projects and pilot projects supported by the TMARC. And the other was to serve as a local and national resource for translational multidisciplinary research and training in drug abuse and neuro-HIV research. So the overarching scientific goal of the TMARC was to define and characterize the combined effects of HIV and meth use on the central nervous system. And then in the renewal period of the TMARC, we shifted our focus to examine the effects of meth and HIV in the context of aging, which converges on similar neural systems and increases risk of cognitive and functional decline. Now, this research is not only important for older individuals with HIV who are current and chronic meth users, but also for studying potential legacy effects of chronic meth use earlier in life that may be more resistant to recovery and are further compounded by aging and aging-related comorbidities. So the overall approach of the TMARC was translational, integrative, and multidisciplinary. And multidisciplinary speaks for itself. We have investigators ranging from basic scientists, infectious disease experts, neuropsychologists, neuroimagers, and so forth. In terms of TMARC's translational approach, this was done by performing studies in both mice and humans, similarly stratified by HIV and meth use, and to the extent possible, using comparable methods to determine effects on brain and behavior. And lastly, the TMARC is integrative in the sense that all cores and projects within the center had synergistic aims and shared methods and data from multiple scientific perspectives to better understand the converging effects of meth and HIV. Next slide, please. So this slide is just a general overview of the TMARC center structure. And like other centers, the TMARC was led by an administrative coordinating core, which directed activities of 3 scientific cores, including the Behavioral Assessment and Medical Core, or the BAM Core, which collected and stored specimens and provided comprehensive clinical data for human participants. The Neuroimaging Core, which conducted human and animal neuroimaging assessments across various modalities. And the Neuroscience and Animal Models Core, or NAM Core, which provided mechanistic insights into the TMARC clinical data using in vivo studies of transgenic mice and human cellular in vitro systems. And the TMARC also included a pilot and developmental core in support of pilot studies and supported 5 scientific projects in animals and humans that spanned various research topics across TMARC scientific themes, as you can see listed there. Next slide, please. So this slide provides just a brief overall summary of the design of the TMARC human component, which used a 2x2 cross-sectional design with participants stratified by meth and HIV status using the criteria shown on the slide. And we try to minimize the incidence of confounding comorbidities with strict medical and substance use exclusion criteria. And again, while the P50 assessments were cross-sectional, we are currently recalling a subset of 200 TMARC participants for comprehensive follow-up assessments, including imaging, as part of an active NIDA-funded R01 that Dr. Latender and I co-lead, which will provide additional resources in support of longitudinal investigations. Next slide, please. So for the sake of time, since many of the clinical assessment resources overlap with those that Bob presented for the HNRC, I'm going to focus specifically on the assessments specific to TMARC and or aging for the next few slides. So this is the first of 2 slides that list resources from the BAM Core's Neurobehavioral Assessment, which again included the neurocognitive battery that Bob described earlier. And also, in addition to composite assessments of complex frontal systems behaviors, including apathy, impulsivity, disinhibition, and sensation seeking. And these were originally chosen for their relevance in our samples and associations with poor decision-making and risky sexual drug use behaviors, which, as you can see, we assessed in our protocol and were central variables for several of the TMARC projects and pilot projects. And we have since found that in some cases, these behaviors remain prevalent in older age, and particularly among our meth group. And lastly, as part of a collaboration between the BAM Core and one of the TMARC projects, we also collected data from experimental measures of assessing aspects of social cognition, including theory of mind, social processing and knowledge, and emotion perception. Next slide, please. So this slide shows again the part of the neurobehavioral assessment resources, and again, many of which were similar to the HNRC, including the psychiatric, mental health, emotional and social functioning and substance use assessments. I do want to emphasize the expanded socio-emotional assessment administered in support of the TMARC Social Cognition Project that administered self-report questionnaires assessing emotional processing, alexithymia, empathy, and attributional bias. And in addition, we also administered additional meth-specific self-report measures to our meth groups, which were administered to further characterize use patterns and behaviors, as well as treatment history. And then again, this was only administered for our meth groups. Next slide, please. So this slide presents resources available from the BAM Core Neuromedical and Laboratory Evaluation, which again is very similar to the HNRC's assessment, so I'm not going to go into too much detail. I do want to highlight the availability of aging-related composite assessments of multimorbidity, functional status, and disease risk, including the VAX index, Framingham risk scores, and frailty indices. And in addition to routine clinical assays, the BAM Core provided indices of biological aging, including cell senescence and mitochondrial damage, as well as a large panel of biomarkers measured in blood and CSF, reflective of inflammation, oxidative stress, and neuronal and vascular injury. And biomarkers of blood-brain barrier integrity and function were also measured in the same TMARC samples as part of my NIDA-funded K23 award that ran in parallel with the TMARC. And in addition to this available data, we have stored specimens which I will summarize at the end of this presentation from the entire TMARC participant group that can be used as a resource for interested investigators. And also, as participants accrue for our R01, the stored specimens will also be available from the follow-up visit for interested groups. Next slide, please. So this is just a brief listing of resources available from the Neuroscience and Animal Models, or NAM Core, and the Neuroimaging Core data. The NAM Core bred and maintained 2 colonies of HIV transgenic mice and examined 2 different meth administration models based on acute and chronic meth use patterns in humans. The NAM Core also conducted neuropathological studies in collaboration with CNTN, which you'll hear about in the next segment of this presentation, as well as in vitro experimental models to examine mechanisms of disease associated with meth and HIV-associated brain injury. The Neuroimaging Core provided cross-species multimodal imaging and provided anatomical, cerebral blood flow, diffusion, and MR spectroscopy imaging data for all eligible TMARC human participants. In the right-hand column, I listed some of the examples of the core mouse and human imaging data that is available as a resource for interested studies. And as with other cores, these investigators are available for consultation when needed with regard to the interpretation of any of the core assessments or any of the data. Next slide, please. So I'm going to finish up with this, and I'll just summarize this real quickly. It's just a summary of the sample size of the cross groups by assessment, and this shows the available stored specimens as well as neuroimaging assessments, and we've broken down the behavioral assessment and the neuroimaging assessment into the numbers for individuals over age 50. And I'm going to skip the rest of the details, but if anyone has any questions regarding this or any of the other data presented, we're going to provide contact information at the end, and I'd be happy to address any questions. So I'll stop here and say thank you for your time, and let my colleague, Dr. David Moore, introduce the resources from the San Diego site of the NNTC.
Wonderful. Thank you, Bob and Jenny, for setting the stage for my presentation. As Jenny mentioned, I'll try not to be redundant with anything that has already been said, but highlight the unique resources that come out of the NNTC and the CNTN. So the NNTC stands for the National NeuroAIDS Tissue Consortium. It's a multi-site collaboration established in the late 1990s. And the really unique difference here as compared to some of the studies you've already heard is that this is set up as a cooperative agreement. It's designed to be a resource and has specifically post-mortem tissues and samples in addition to in-life assessments. So it's based on 4 clinical sites that gather data, one at the University of Texas Medical Center, led by Ben Gelman, the CNTN site in California and San Diego, led by myself and Chris Akeem, the NNAB at UCLA, led by Elise Singer, and then the Manhattan Bank, led by Susan Morgello in New York. And there's a data coordinating center that's led by University of Nebraska and Howard Fox, as well as MS Corporation. Next slide, please. And so what we have in this cohort, the NNTC cohort, is we have just under 1,000 cases that have both in life as well as, I should say, these are cases with postmortem tissues of brain samples available for research, just under 1,000 HIV positive cases and 267 HIV negative cases. In addition to those cases that have brain available, the great majority of them also have other tissue samples from other organs, liver, spleen, kidney, heart, and peripheral nerve samples that people can relate to the brain tissues as well. We also gather in-life fluid, plasma, serum, PBMCs, and CSF that can be analyzed and gather those at the time of death as well, where possible. And then we've followed extensively with similar approaches already seen in the HNRC and TMARC for neuromedical, neuropsychological, and psychiatric data. And importantly, there's a subset of individuals that's unique and important that have been virally suppressed over long periods of time, but yet still have either cognitive problems in some cases and have all of these data available. And on the nntc.org website, there's a query tool that people can look and see what cases might be matching their research questions. Next slide, please. So this is a very busy slide, but in general, if you orient you to the top most box, we've enrolled over 3,000 participants into this study. The great majority of those following the branch to the left have been people living with HIV. If you follow out to the far left side, you can see that there's been almost 600 people who've had an autopsy in study in the lower left green box. And then if you follow also the green down to the bottom, the currently active cohort, we have 579 currently active people. And of those, 515 are longitudinal evaluations, meaning they have at least 2 visits on study and are continuing to be followed. So we have a really rich resource of people, not only who have had in-life assessments and then come to autopsy, but also people who are actively being studied as well. And then we have a smaller subset, but still some currently active and comparison cases who are not living with HIV. Next slide, please. And this slide shows just our accrual over time, starting at 2001 on the x-axis through 2018, the most recent year where we've compiled these data. And again, you can see that there's over 1,000 people deceased with autopsy, and then our active cohorts each year are about around 600 people. Next slide, please. And this shows that just over time, the persons in our research studies are traditionally getting much older. So over half of our cohort, which is the great majority of these people are living with HIV, are 60 years old or older, and another third or so are 50 years of age or older. And you can see how that has changed from the early 2000s to now, where we rarely had anyone over age 60, to now where our cohort 20 years later is the majority are over age 60. So this really is an aging study now of people aging with HIV. Next study. Sorry, next slide, thank you. So, and then the assessments are very similar to the HNRC assessments that Dr. Heaton mentioned earlier, and in fact are probably a little bit of a subset of those, but we have very detailed neuromedical assessments, as you can see, some of the domains listed here and more detailed in the HNRC, neuropsychological assessments covering the 7 domains that Dr. Heaton mentioned earlier, as well as daily functioning and sleep quality, and then labs, again, consistent with the HNRC, as well as psychiatric evaluations, again, consistent with the HNRC. Next slide, please. And again, the unique part of the NNTC is this ability to look at the in-life assessments listed under the clinical examination box that cover neuropsychological, psychiatric substance use, neuromedical, and in-life fluid sample collection, blood and CSF, to what happens in individuals who come to autopsy and we're able to get tissue samples of brain and other organs and look at relationships between what was going on in life and their pathology after death. And this is a really unique resource, and people can request both data from the resource as well as fluids, as well as the tissue samples themselves, which can be both fixed and frozen. Again, a really unique resource, and is set up to be a resource such that other people can get these data and samples. Next slide, please. So just to give you a sense of some of the publications that have come out of this study, a couple recent ones from the last couple years. Dr. Morgella in New York led a study that showed neuropsychiatric illnesses, particularly symptomatic cognitive impairment and depression, were associated with increased frailty risk, and a lot of that was driven by both walking speed and grip strength in people who were symptomatic with HIV, so showing the use of our cohort. And then a subsequent or a different study led by Aaron Sunderman, assistant professor in our group, looking at inflammation-related genes and how they may be associated with accelerated aging as measured by the epigenetic clock of Steve Horvath. So these are some of the in-life assessment modalities that we can use to be able to find interesting findings with this large multi-site cohort. Next slide, please. And this is a descriptive study that just talks about what the NNTC is, how the database is structured, and gives some insight into how the platform is set up for data management, and gives you some insight into the multi-site nature of it. So important to highlight. Next slide, please. So I'm going to shift gears here from the unique resource of the NNTC again, which has the pathologic samples, which is really the unique part of that study, to a recently completed R01 study that I did with Dilip Jesti at UCSD and the HNRP. And this really tried to focus on successful aging among people with HIV. Next slide, please. In this NIMH-funded study, we had a what's called a multi-cohort longitudinal design, such that we took people in 3 pre-specified age bins or cohorts. So we had in the people living with HIV, we had 40 individuals in each of 3 age bins, 36 to 45 years old, 46 to 55 years old, and 56 to 65 years old. And we had a comparable HIV negative cohort, 30 individuals in each of those 3 groups. And so this allows really for direct age cohort comparisons over time. And our study schema was to get comprehensive assessments, again, consistent for the most part with the HNRC battery that Bob described earlier, to get those comprehensive assessments in years 1, 3, and 5, and then to get some specific questionnaire data in years 2 and 4. And I'll talk about what those data are on the next slide, please. So we tried to get both physical, cognitive, and psychosocial functioning data, as well as self-rated or self-perceived successful aging data. What's really unique to this study is the positive psychological factors and that self-perceived successful aging factor, which is why they have asterisks here. And I'll talk about those in more detail in the next slide. We also get systematic biomarkers in that study as well. So here, thank you for going back, and some key covariates, psychiatric and substance use data. Next slide. Some of the other unique things that we gather in the successful aging study are attitudes and opinions about aging. We get some data on religious and spiritual views, sex life, and some reflection on the individual's life as well. Next slide, please. And really, one of the most unique parts of this study is this focus on positive psychological factors. And I won't go through each of these in detail. People can go back and look at these scales later on, or please follow up with me if you would like to learn more about what we've done or for opportunities to work with these data. But we covered domains such as grit, so the ability to bounce back when things are difficult, social support, resilience, optimism, life satisfaction, personal mastery, geriatric morale, and then some not positive ones, but the inverse of them being anxiety and perceived stress. But really focusing on these top ones and how they may be able to buffer some of the potentially negative effects of living with HIV was one of the main goals of this study. Next slide, please. So example publications that we've had here, looking at frailty and seeing that having a positive outlook on life really may help buffer people from developing frailty. A second was that looking at depressive symptoms as a way to being associated with health-related quality of life, and the absence of certain positive psychological factors. Again, speaking to the idea that if we're able to buffer certain depressive symptoms through positive psychological factors, we may be able to make changes. Next slide, please. So I'm going to hand it off to my colleague, Scott Latender, to finish the San Diego related resources, and he'll also take questions at the end. So all yours, Scott.
Thank you, David, and thank you, Bob and Jenny. Also, all the nice data that everyone presented makes my job much easier because there are a lot of similarities between these projects. The Charter project is a bit different, though, in that, like NNTC, it's a multi-site project. And you'll notice that the NNTC has sites in Texas and Manhattan, and those same sites are also Charter sites along with San Diego. And but to those sites, we added sites at Johns Hopkins, the University of Washington, and Washington University in St. Louis. Charter started nearly 20 years ago, and the goal was to assess 1,600 people with HIV and to follow 600 of them over time, initially every 6 months, and then over time this changed to 1 year, every 2 annual evaluations. Next slide. So this intends just to summarize the rather complex history of Charter, the cross-sectional component of the study with the 1,600 people. The longitudinal phase, we ended up adding some people with early HIV infection and other conditions of interest, so that 600 grew to 700 over time, and then declined in what was called the option period to 300 and then to 180. And currently we are completing the Charter aging study, in which we brought people back after 12 years, about 400 people after 12 years after their initial evaluation. Next slide. So Charter has accumulated valuable clinical and research data from these people, longitudinal data from the 700, and then, and similar to our other studies, we collect neuromedical data, neurobehavioral data. There's an imaging component to Charter as well. And we also have viral genetics in a subgroup, as well as host genetics. We performed a genome-wide association study in about 1,100 of the 1,600 people in the cross-sectional cohort. The biospecimen repository currently houses over 100,000 aliquots of specimens, and we have over 5 million data points. Next slide. So this is just similar to tables that were in the other parts of the presentation, and summarizes the stored data. So again, neurobehavioral characterization, neuromedical characterization. We collect data on psychiatric disease and substance use disorders, blood biomarkers such as clinical chemistry panels, viral load, CD4, and so on. And then another distinctive element of many of our studies, including Charter, is that we collect CSF. So Charter is perhaps the most successful of our studies in the in successfully getting lumbar punctures, performing lumbar punctures. We had nearly 80% of the 1,600 participants successfully undergo a lumbar puncture. And while CSF has been used in many of those participants, we have CSF stored also for the majority still. Next slide. This is in the longitudinal component. Again, we have over 700 people, and this slide simply summarizes the number of participant visits for each of these types of assessments. In addition to structural MRI, we have magnetic resonance spectroscopy. We also performed quantitative sensory testing and neuroconduction velocity, as well as skin biopsy for intraepidermal nerve fiber densities. And that's because not only were we interested in these issues, and they're important in people with HIV, but one of the funding agencies was the National Institute on Neurological Disorders and Stroke, in addition to the National Institute of Mental Health. And then you see the number there from people who are older than 50 years. Next slide. This table, again, is just a summary table that you can refer back to when you download the slides, but summarizes the types of biospecimens that we have in storage currently. You'll notice, in addition to CSF-derived and blood-derived specimens, we also have buccal swabs and saliva. We have PAX gene RNA and DNA tubes, and we've also worked with our collaborators to extract DNA in known concentrations to better enable future genetic studies. Next slide. So This doesn't differ greatly from slides that you've already seen for the other projects, so I won't read through these lists. We get neuromedical data and laboratory data. One distinction here is two distinctions I'll mention again is the host genotyping, but also the antiretroviral drug concentrations. Pharmacology was a particular focus of Charter. One of the main aims of Charter was to understand how antiretroviral therapy was affecting people with HIV because it started in 2003, relatively early in the HART era. And so we also have antiretroviral drug concentrations stored from blood and CSF for several antiretrovirals. Next slide. And I mentioned the genetics data. So we have the genome-wide association study in 1,050 people. We also included mitochondrial genotyping and haplotypes, working with Todd Holgan and David Samuels at Vanderbilt, as well as HLA typing and KIR typing, working with Sunil Ahuja at University of Texas, San Antonio. And from these data, they've also estimated genetic ancestry. So we have not only the self-reported ancestry on people such as African ancestry or European ancestry, but actually the computed ancestry from the GWAS project. Next slide. So these are additional Charter resources from the neurobehavioral data and the neuroimaging data. Again, I won't read off these lists, but they're similar to what you've seen in the other projects. Next slide. So in summary, the HNRP at UC San Diego can provide a substantial volume of data and biospecimens in support of research on HIV and aging, with particular focuses on the brain and addiction. Data and biospecimens can be requested by completing request forms available from UC San Diego, and you see our website there, or from the National NeuroAIDS Tissue Consortium. We also lead developmental grant and training programs, including the Sustained Training in Aging and HIV Research Program, which is led by Dilip Jesti and Maria Marquine and myself. Next slide. And if you're interested in requesting resources, you can either go to our website, as I mentioned, or you can email hnrpresource@ucsd.edu. Part of our mission, as Bob mentioned, is to support new research in neuro-HIV and aging. And so we're very happy to answer questions and to be engaged about research projects. So if you want to email us directly as well, here are our email addresses. So next slide. The next component is to ask for questions, and just to remind everyone, we'll not use the raise hand feature today for questions, so please use the questions feature that's accessible on the right side of your screen. And if we do not get all the questions today, we'll email responses after the webinar. So Judy has made me an organizer so that I can see questions that are typed into the question box, but right now I don't see any. Among the attendees, I see some of our STAR scholars, the Sustained Training in Aging and HIV Research. So not putting anyone on the spot, but I see Alexis there, and Dr. Perkins from who's not one of our trainees. She's one of our mentors and faculty, but Dr. Perkins from Emory as well. So there is a question. How is adherence measured in these studies? So let's see, can I open that up to our panel and see if there's anyone who wants to volunteer to respond to that?
Well, you're very familiar with the ACTG self-report. ART adherence, right?
I am, yes.
Right. So you want to just go ahead and describe that?
Okay.
We also assess for detectable virus in blood and CSF. That doesn't detectability doesn't mean that they're not adherent, but we have those two ways of looking at it, one objective and one subjective.
So Bob mentioned the adherence, the ACDG adherence questionnaire, which asked participants to self-report how many doses they've missed in the last 4 days. And we use that because it's standardized, it's straightforward, and it was shown back in the early 2000s that people who took more than 95% of their doses over 4 days were more likely to have a suppressed viral load. And people who took fewer doses, who missed doses more than that, were more likely to have detectable viral loads. So it was a well-done study. The environment certainly is different now than it was back in the early 2000s with regard to antiretroviral therapy. But because we've been using that assessment for 15 to 20 years, we continue to use that. As Bob said, you can also look at the viral loads. And as I mentioned in some of our studies, we measure antiretroviral drug concentrations. And while antiretroviral drug concentrations are important, it's important to interpret them in the context of the time that the specimen was collected after the dose, and other factors like age and sex and food intake and so on. If the drug concentration is 0 after someone's taken a dose or reports taking a dose, then that's a fairly good indication that non-adherence may be present. And so for the substantial majority of people, we do not have drug concentrations on them, so we rely on the ACTG adherence questionnaire. So Dr. Bender says, gives thanks to the panel for the presentation, but doesn't have any questions. Jennifer Kirk, so I didn't mention the name of Xiaoying Yu, who asked the adherence question. So I'm mentioning other people's names. So Dr. Yu, thank you. So Jennifer Kirk, you mentioned that meth and HIV can affect executive function. Can you clarify the additive impact on the brain? Is this general damage or increasing white matter ADRD risk? So Dr. Udicello, do you want to respond to that?
Sure. Sorry, I was trying to get to my audio. The research that we've been doing does show additive effects on cognition, but we are seeing that meth seems to behave a little bit differently in the HIV positive individuals. So that's a group that we're still trying to explore. We think maybe that it might have to do with differing meth use patterns, but we do see additive neuronal damage. The white matter is actually follows a similar pattern to the cognition. So again, we've been focusing a lot lately on looking at this dual risk group to see what characteristics of what individual characteristics and like demographics or meth use characteristics or other substance use characteristics may be driving the individuals who actually show greater damage versus those that show less. I don't know if anybody else wanted to add something else.
Dr. Kirk, do you have any follow-up questions for that? While you consider that, let me just respond to a follow-up question from Dr. Yu. Dr. Yu asked what the justification is for using a 95% versus the 90% cutoff. So I may have misspoken. We collect just how many doses they miss, so we can look at 90% or 95%. And so if the original ACDG assessment was 90%, I may have just misremembered that. So was not intended to take a stance on one versus the other. In fact, with longer acting therapies now, I think that perhaps people have to miss more doses in order to have an effect on their viral load and development of resistance than back when the therapy had to be dosed 2 or 3 times a day. So, all right. Well,
so Scott, I think you know, some of the takeaway messages that we might just want to reiterate here are that, so we showed you 5 different research projects that have a multitude of data and samples, and that we are in some cases set up to be resources for those studies. So we have an explicit aim to be sharing those data and resources with other individuals who are interested in them. So we're designed to help and collaborate. So people should feel empowered to reach out to us and make use of those data and resources. So that's one takeaway. A second takeaway is that whether there were explicit aims in each study to study aging or not, and many of them did have explicit aims to look at relationships of aging with HIV, our cohorts and individuals with HIV are increasingly getting older and are in older age bins. So that and there's a lot of open questions about how aging affects people who are living with HIV. So I think that there are many open questions, and we have a lot of resources and data and samples that people can capitalize on. So that would be just 2 takeaways for all the attendees.
Thank you, Jenny, Bob, Scott, and David. It's been a real pleasure learning about the UCSD and collaborating sites data sources. Please know a webinar survey will automatically launch after the program. In an effort for continual improvement, we would like to hear your thoughts. Please provide feedback by clicking the survey link at the end of the webinar. And I'll also share and ask and invite the audience to please join GSA on November 4 to 7 at the GSA Reimagined 2020 Annual Scientific Meeting online. The meeting will be hosted on a state-of-the-art platform that will offer an immersive experience, allowing you to focus on essential research and networking. Thank you to everyone for participating. This is the end of the program.